This
Let us
conference will
begin.
now be recorded.
So the contents you will be learning under the topic alkaloids are introduction, its history, occurrence, definition, properties, classification, isolation and extraction, chemical tests of alkaloids, and uses of it.
Sejal,
Introduction.
just a second. Sejal, 1 second.
Yes, yes ma'am. Yes, ma'am.
Yeah, all others, I want all of you to note down, okay? You have to write down today's date, write down the topic, and all keywords like biological source, family, chemical constants, uses, all keywords has to be written. Whatever doubts you get, it has to be written as question. And after the presentation, for 2 minutes, question and answer session will be there. So you're going to ask your questions. All of you should write this in your book. Clear? Yes, Sejal, you can continue.
Introduction. Alkaloids are a heterogeneous group of natural substances having more than 6,000 basic nitrogen-containing organic compounds. That means alkaloids are very vast and have more than 6,000 basic nitrogen-containing organic compounds. These are mainly helpful in the chemical, physiological, taxonomic, and biogenetic studies. Let us study the history of alkaloids. The term alkaloid or Pflanzenklein was coined by Messner, who is a German scientist, in 1819, followed by Dawson in 1803 isolated narcotin, and in the same year morphine from opium was isolated by Saturner, followed by Pelletner and Calverter isolated emetine in 1817 and colchicine in 1819. The first alkaloid for its structure to be known was quinine in 1826. Then, by 1884, about 25 alkaloids were isolated from the cinchona bark alone. In 1889, quinine became the first synthesized alkaloid. As we have seen earlier, it was the first alkaloid for its structure to be known, and later on it became the first alkaloid to be synthesized. In the present century, proper structures were assigned to many of the alkaloids with the help of chromatographic and sophisticated physical methods of analysis. Now, let us study about the occurrence. So, as we all know that alkaloids are present in most of the natural occurring substances. So, we find majorly these in the angiosperms, which are flowering plants. So their presence is detected in micromarine organisms, insects, animals, and lower plants. Some of the alkaloids from animal kingdom are castorenin from Canadian beaver, muscopyridine from musk deer. Most alkaloids occur in the skin of amphibians along with other toxins. Out of 60 different orders in higher plants, 34 orders contain alkaloids. That means more than half of the orders have alkaloids in them. And some of the families which contain alkaloids are Apocynaceae, Liliaceae, Papaveraceae, Solanaceae, and many others. The alkaloids are present in morphological parts of the plant. Example, in seeds such as in Nux vomica plant, roots of Rauwolfia, underground stems of Sanguinariae, in the barks of cinchona, in the leaves of coca leaf, and in fruits such as in conium. In some cases, like vinca and ephedra, practically every part of the plant contains alkaloid. That means the whole plant majorly contains alkaloids within them. Let us study the definition of alkaloids. The term alkaloid is derived from the word alkali-like. That means which resembles some characters of naturally occurring complex amines. As we all know the definition of alkaloids, these are organic compounds, products of natural or synthetic origin, which are basic in nature and contain one or more nitrogen atoms, normally heterocyclic of nature, and possess physiological action on human and animal body when used in small quantities. These are highly toxic in nature and poisonous in nature, but if used in small quantities, These show a very useful physiological effect. Properties of alkaloids. Physical properties. Many of the alkaloids are colorless, crystalline solids with having sharp melting point. Some alkaloids are amorphous gums, while others like quinine, spatine, and nicotine are liquid and volatile in nature. Some of the alkaloids are colored in nature. Example, beta-adenine is red, berberine is yellow, and salts of sanguinarine are copper red in color. The free bases of alkaloids are soluble in organic nonpolar or immiscible solvents. The salts of most alkaloids are soluble in water, but their free bases are insoluble, and their salts are very sparingly soluble in organic solvents. The alkaloids containing quaternary bases are only water soluble. Some of the pseudo and proto-alkaloids are highly water soluble. Example, colchicine is soluble in alkali water, acid, or water. Caffeine, like the free base of caffeine, is freely soluble in water. Quinine hydrochloride is highly soluble in water. Now let us study the chemical properties of alkaloids. These are basic in nature due to the availability of lone pair of electrons on nitrogen. The basicity of alkaloids is enhanced if the adjacent functional group are electron releasing. That means if the functional group are electron releasing, the basicity is enhanced. And if you want it to be neutral or acidic, then the functional group should be electron withdrawing. The alkaloids may contain one or more number of nitrogen, and it may exist in the form as primary, secondary, tertiary, and quaternary ammonium compounds. Example for primary amine is mescaline, for secondary amine is ephedrine, tertiary amine is atropine, and for quaternary ammonium compounds is tubercuranine chloride. In the natural form, the alkaloids exist either in free state as in amine or as salt with acid or alkaloid. Now let us study the classification of alkaloids. These are first classified on the presence of nitrogen as true alkaloids, proto-alkaloids, and pseudo-alkaloids. True alkaloids. These are toxic in nature and are basic in nature. These contain heterocyclic nitrogen, which is derived from amino acids. Proto-alkaloids, also known as amino acids, these are simple amines and basic in nature. In these proto-alkaloids, the nitrogen is not present in the heterocyclic ring, but it is present in the side chain. Example, mescaline, ephedrine, N-common dimethyltryptamine. Pseudo-alkaloids. These are not derived from amino acids. Neither they show many of the typical characters of alkaloids, but they give standard qualitative tests for alkaloids. Example, carnosine and caffeine. The second classification is based on the pharmacological classification. The alkaloids are classified under various categories like CNS stimulant, depressant, sympathomimetics, analgesic, purgatives, etc. Within the same drug, the individual alkaloid may exhibit different action. Example is opium. In opium, morphine and codeine both are present. So the morphine is narcotic analgesic, while codeine shows antitussive. Antitussive is mainly used, means it is used in the relief of cough. And another example is in cinchona, quinine is anti-malarial, while quinidine is cardiac depressant. The third classification is based on the taxonomy. Uh, Taxon means it is based on the family. The classification is based on the family. Some examples are cannabinaceous alkaloids, example hemp and marijuana. Rubiaceous alkaloids, example quinine, yohimbe. Solanaceous alkaloids, example benadonna, ashwagandha, and many other like apocynaceae, liliaceae. Many other categories are classified under this. The fourth classification is based on the biosynthetic classification. In this, mainly the significance is due to the precursor from which the alkaloids are biosynthesized. Precursor is nothing but the starting material from which the alkaloids are synthesized. So if the drugs having different taxonomy and physiological activity, yet they can be grouped under the same biosynthetic classification if they have the same precursor. Then the alkaloids are categorized on the fact whether they are derived from amino acid precursor as ornithine, lysine, tyrosine, phenylalanine, tryptophan, etc. The last classification of alkaloids is the chemical classification, in which these are classified on the basis of heterocyclic structure present in the alkaloid. The chemical classification of alkaloids is based on two types, heterocyclic alkaloids and non-heterocyclic alkaloids. Heterocyclic alkaloids are also called as true alkaloids. These are further divided into 12 groups according to the nature of their heterocyclic ring. And non-heterocyclic alkaloids are also called as proto or pseudo alkaloids. Now let us study about the heterocyclic alkaloids, which are divided into 12 groups. The first one is pyrrole and pyrrolidine. The structures are given. Example which comes under this group of classification is hygrine and coca species. Next, pyridine and piperidine. Example, quinine and lobelin. Third one is pyrrolizidine. Example is echimidine and symphatine. The fourth one is tropane. Example of tropane drugs are atropine, hyoscine, and cocaine. Next, quinoline. Example, quinine and quinidine. Isoquinoline, emitin, narcotin. Phenanthrene, morphine, codeine. Aporphine, baldine. Indole, brucine, physostigmine. Imidazole is pilocarpine, isopilocarpine. Norlupinane is patine and cysteine. Cytisine. Purine is caffeine and theobromine. So these were the classification, that is 12 groups, which comes under heterocyclic compounds. Now non-heterocyclic alkaloids. First one is pseudo-alkaloid, that is diterpene. Example is aconitine, aconine. Next, proto-alkaloids under this alkyl amine, which is also amino alkaloid. Example is ephedrine and mescaline. Now let us study about the isolation and extraction of alkaloids. The extraction of alkaloids is based on their basicity and solubility pattern. We'll study two methods of isolation and extraction. So first one is the Morstern drug is treated with alkali so that the base is free from this, and as it exists in the salt form. The Morstern drug actually exists in the salt form. So we treat it with alkali so that the base is free. Then to separate free base with polar solvent. This is known as Stas-Otto method. The alkaloidal salt is transferred to polar solvent, followed by the aqueous phase is made alkali with sodium carbonate or ammonia, and then extracted with organic solvent, followed by drying with sodium sulfate, filtered and evaporated to yield alkaloidal residue. I'll repeat. The Morstern drug, which is in the salt form, is treated with alkali so as to set free the base. Then this is treated with polar solvent. This is known as Stas-Otto method. The aqueous phase is made alkali with sodium carbonate or ammonia, followed by extraction with organic solvent, any convenient organic solvent. Then the drying of alkaloids is with sodium sulfate and filtered and evaporated to yield the alkaloid residue. The other method is to treat the drug with ammonia, so it converts the alkaloidal salt to their free bases, followed by the liberated alkaloids are extracted with organic solvents like ether, benzene, chloroform, etc., and the alkaloidal residue is obtained. These can be further purified by any of the process given below as crystallization, steam distillation, chromatography, and gradient pH technique. This is a schematic representation of isolation and extraction of alkaloids. The powdered drug, which contains alkaloids, is treated with sodium carbonate, ammonia, calcium hydroxide, and the alkaloids are freed as bases. Then these exhaust with organic solvent as chloroform ether, and the total extract is formed. This extract concentrate and shaken with successive quantities of inorganic acid. Two types of solutions are formed: residual organic fraction and aqueous acid solution, and followed by at the end we get residual alkaloid. Chemical test of alkaloids. There are basically four chemical tests for alkaloids, which are Dragonroth's reagent test, Mayer's reagent test, Wagner's reagent test, and Hager's reagent test. Dragonroth reagent test consists of potassium bismuth iodide solution, which gives brown or reddish brown precipitate when treated with the sample. Mayer's reagent consists of potassium mercuric iodide solution, which when treated with the sample gives cream precipitate. Wagner's reagent consists of iodine and potassium iodide solution, which on treatment with the sample gives brick red precipitate. Hager's reagent, which consists of saturated solution of picric acid, on treatment with the sample gives yellow precipitate. So these were the four chemical tests for alkaloids. Uses of alkaloids. As earlier said, that alkaloids are poisonous in nature, but if used in small quantities, they exert useful physiological effects on animals and human beings. These act as protective against insects and herbivores due to their bitterness and toxicity. They even act as a final product of detoxification of waste materials. They act as a source of nitrogen in case of nitrogen deficiency, and also as growth regulators in certain metabolic systems. They may be utilized as a source of energy in case of deficiency in carbon dioxide assimilation. Alkaloids act as local anesthetics. Example, cocaine acts as amebocides, as emetin, as antibacterial, berberin, as antitumor, vinblastine, anti-malarial quinine, and as painkiller, we already know that it is morphine. Now let us study briefly about the drugs which comes under the class of alkaloids. Four drugs are given in the syllabus: vinca, rauwolfia, belladonna, and opium. So let us study about the vinca. The synonyms for vinca are catharanthus and periwinkle. We have all seen this flower in our surroundings. So this is basically, it occurs in four colors also: white, pink, red, and violetish. So the biological source of vinca is, it is the dried whole plant of Catharanthus roseus. It is also known as Vinca rosea. The family to which vinca belongs to is Apocynaceae. Uses. Uses of vinca are, it is used as an anti-neoplastic agent which may act by arresting mitosis at metaphase or by interfering with amino acid metabolism. It also exhibits hypotensive and anti-diabetic action. The second drug under alkaloids are rauwolfia. Synonyms are rauwolfia root, serpentina root, chota chand, and sarpagandha. The biological source is, it consists of dry roots of plant known as Rauwolfia serpentina. Sarpagandha contains not less than 0.15% of reserpine and ajmalicin. The family to which Rauwolfia belongs to is also Apocynaceae, as earlier seen in vinca. The uses are, it is used as an antihypertensive drug. Then the drugs under which consist of Rauwolfia, that is reserpine, it treats mild to severe hypertension. Desirpidine is used as antihypertensive and tranquilizer. Agmalicin is used in the treatment of circulatory diseases. Belladonna. The synonyms for belladonna are belladonna leaf, belladonna folium, deadly nightshade leaf. The biological source is it consists of dried leaves or other aerial parts of Atropa belladonna lin. Lin is the name of the scientist, or Atropa acuminata. It contains not less than 0.3% of the alkaloids of belladonna herb. The family to which belladonna belongs to is Solanaceae. The uses of belladonna are it is used to reduce the secretions such as sweat, saliva, gastric juice, and also to reduce spasms in case of intestinal gripping due to strong purgatives. It is also used as an antidote in opium and chloral hydrate poisoning. Opium. The synonym for opium is raw opium. The biological source is it is a dried latex obtained by incision from the unripe capsules of Papaver somniferum lin. It contains not less than 10% of morphine and not less than 2% of codeine. The family to which opium belongs to is Papaveraceae. Uses are this mainly belongs to the category of sedative hypnotics, and the main analgesic activity is due to morphine. Morphine is a potent analgesic. Codeine acts as anti-irritant in the bronchial tract and then as antitussive in cough remedies, that is, in relief of cough. Papaverine acts as smooth muscle relaxant in the intestinal tract. Thank you.
Yes, Sejal, very good presentation. The flow of the schematic thing was also very good. I hope everybody has understood. In case you have any doubts, you can please ask Sejal. And one, Sejal, in this purification, you've told about crystallization and other three things.
Yes, ma'am.
Just add fractional liberation also in that.
Okay, ma'am.
Okay, remember we had discussed that alkaloids are different pH, different alkaloids comes out. Like at one particular pH, primary alkaloids will come out, at another pH, secondary alkaloids will come out. Similarly, tertiary and quaternary. So fractional liberation is one technique where after isolation, you can purify and collect different type of alkaloids. So add that in your slide.
Okay, ma'am, will do.
Before circulating. Now, what I want to tell is, I'll be sending a link of this class. Now, Sejal, you have presented, right? So what you can do is, you can fill in that form. It has got you to fill your details like your name, your USN number, your class, and your PPT should be uploaded there. Okay? And make a notes of this, whatever PPT you're presenting, a handwritten notes, you can just prepare and upload it there. By evening, you can upload it. Clear?
Okay, ma'am.
Okay. Then what else I want all of you to do is, all of you listen, whichever topic you're presenting. So you read it thoroughly, understand it, make 10 MCQs out of that. Okay. So whatever you're presenting, you would be having the matter with you. Like for example, in alkaloids, what you can give is one question, which is the method of isolation of Stas-Otto. Stas-Otto is the answer. So what you can do is some 4 similar names or some 4 different things. Like for example, you can give enfleurage, usilly, maceration, and Stas-Otto. So Stas-Otto method will be the answer. Make just 10 MCQs for each of your topic. Clear? And 2 or 3 subjective type questions. Is it clear?
Yes, ma'am.
Yeah. Sejal, did you understand what I said you?
Yes, ma'am, understood.
Yeah. What you can do is, I think all of you are familiar. I mean, you know how to create Google Forms. If not, also you can just go to your Gmail account. Okay. In that you can go for Google Drive. You can go to Drive, and there you can go for Google Forms. Is it clear? So there, you can just start putting your questions there, and 4 options you can give, and there itself you can give the right answer. If you want, you can give the explanation for the right answer also. So you can choose one attempt. So when you click this, when you pass on this link to your classmates, they'll be able to answer once. So you would have allotted marks in that. So system itself will analyze it. Is it clear? In this class, who has done Google Forms as of now for any purpose? Yes, Mahantesh?
No, ma'am, I'm not done.
Today what you can do, no, just try it with 2 or 3 questions. It is easy. Go to the drive and there you can select blank form. Just give the title, whatever your assignment topic is, you can give the title for that, and you can give subjective or objective. Okay, and there you have in your right side, no, you'll get some 4 options, which type of question you want, whether MCQ or bulleted or how you want. You can select MCQ based. So just type in the question and try this. Who is very good in computer and all in your class? Mahantesh, is it clear?
Yes, ma'am.
So Sejal, you try this. After this, you try. In case you're having any doubts, no, you can call me.
Yes, ma'am. Okay.
Okay, done. So anybody else has any questions, you can ask Sejal. Come on, I want at least 1 or 2 doubts from your side. Or Sejal, you can ask, you can call any student and you can ask what they have understood, or whatever you've explained, you can just ask them to answer it. This 1 minute or 2 minutes of question answer discussion should be there for all PPTs. If anybody has any question, you can ask. Otherwise, Sijal will be asking you the question. Sijal?
Yes, ma'am.
Yeah, you can ask 1 or 2 questions to anyone. You can just tell roll number. There's a unbiased thing here. No need to call names. You can just tell some roll number and just ask them any small questions with whatever you have covered. I hope all of them have understood, because this was also very clearly discussed in theory class. So maybe in this topic nobody has doubts, but others you should start getting doubts. Sijal, 1 or 2 questions you can pose and end your session.
I'll just ask the question, ma'am. Whoever knows can answer.
Nobody will, you see, like me, you will be only shouting answer, answer, answer. You'll be telling nobody's going to answer. But if 1 or 2 answers, fine. Otherwise, we will ask the roll number. Or you ask the question, I'll tell the roll number, let them answer.
Okay, ma'am. So, what are the chemical tests for alkaloids?
See, such a easy question she's asked. She don't want to put her classmates in difficulty. Roll number 17 can answer this. Who is roll number 17? Let me just check. Gokul Gautam. Now, for answering the questions also, definitely you will have points which will be added to your assignments. If Gokul Gautam is not answering, it will be -1 then for him. Yes, anyone else can answer this question? Miss
Dragonrock
Bharti.
test, Hackers test,
Okay.
Mayer's test.
Yes.
That's it, ma'am.
One more is Wagner's. Wagner's. Very good.
Wagner's.
Very good for your
Wagner's.
attempt, Bharti. I want all of you to, involve yourself and what happened? I want all of you to involve yourself and answer like this. Let it be an interactive session. Any other questions, Sejal?
Classification based on the presence of nitrogen.
Yeah. So she's asking, how do you classify alkaloids based on the nitrogen?
True alkaloids, protoalkaloids, and pseudoalkaloids.
Very good. So Chaitra and Bharti will be scoring extra 1 point today. Fine. So Sejal, thank you. Very good presentation. We'll start with the next person. Shilpa, who's the next person?
Miss Shagupta, ma'am.
Shagupta, you're there?
Ready with Vinka.
Yes, ma'am.
Shagupta, you're ready? You're ready? I'll make you the presenter. You can share your screen.
Yes, ma'am.
What have you joined? Your name is not seen in the attendees list. Okay, 1 second. Yeah, I've made you the presenter. You can join. You can share.
Yes, ma'am. Ma'am, how to share? Excuse me, ma'am.
Next to the mic button, you have a share option. You can just click on that and share.
Okay. Good morning to all. My topic is Vinca.
Yes, good afternoon. You can start.
Good afternoon. Sorry. The topics which I am going to cover under these topics are synonym, biological source, geographical source, collection, cultivation, and preparation for the market, macroscopic characters and microscopic characters of Vinca, chemical constituents, uses, doses, and last is commercial products. Vinca belongs to the family, to the anti-tumor category, that is the drugs which are used to treat cancer. The synonym is Catharanthus periwinkle, Vinca rosea, and in Hindi it is also called as Sadabahar. The biological source of Vinca is it is the whole dried whole plant of Catharanthus roseus belonging to the family Apocynaceae. Next, geographical source. It is probably indigenous to Madagascar. It is cultivated in South Africa, India, USA, Europe, Australia, and Caribbean islands as an ornamental plant as well as for its medicinal properties. Next topic is collection, cultivation, and preparation for the market. Vinca grows all over the India. It is perennial and retains its glossy leaves throughout the winter. It is grown well in tropical and subtropical area in South India and northeastern states of India. It favorably grows in light sandy soil which is rich in humus. The rainfall of about 100 centimeter is most favorable for it. It is cultivated by two types, either by directly sowing the seeds or sowing the seeds in the nursery. For direct sowing, about 2.5 kg of seeds per hectare is required. Nursery sowing method is found to be economical, and the fresh seeds are sown in the month of February or March. This graph gives the information about the collection, cultivation, and the preparation of Vinca. After sowing the seeds, after 2 months, they will achieve a height of 6 to 7 centimeter, and later they are transplanted to the open fields. The plants The plant does not require any special supply of fertilizer, as the mixture of nitrogen, phosphorus and potassium gives favorable results. Proper fertilization and weeding is done timely, and leaf stripping are done 6 to 9 months after sowing. The stems are cut about 7 to 8 centimeter above the ground level, and the leaves, stem and seeds are separated and air dried. The roots are dug out by plowing, which are further washed, dried in shades and packed in bales. The seeds are collected from the mature fruits of vinca for the next propagation. The plant contains less percentage of alkaloid, hence it is not used as a galenical but is used for the extraction of alkaloids. The alcohol extraction is based on the solubility and insolubility in different solvents. Due to this, vincristine, vinblastine and other weak bases are separated and then fractionated with the help of column chromatography using aluminum as adsorbent. By using tissue culture technique, some of the vinca dimeric alkaloids like catharanthine and ajmaline are isolated. Next is macroscopic characters. As in the picture we can see, the leaves are green in color, roots are pale grey, flowers are either violet, pinkish white or carmine red in color. Odor is characteristic and taste is bitter. Vinca is an erect, pubescent herb with branched taproot. Leaves are simple, petiolate, ovate or oblong, unicostate, reticulate, brittle with acute effects and glossy appearance. Flowers are pedicellate, complete, her, hermaphrodite, normally 2 to 3 centimeter in cymose axillary clusters. Hermaphrodite means thing which has both male and female organs. The fruits are follicles with several black seeds. Normally we find three types of vinca plants. First is Vinca alba, which has white color flowers. Second is Vinca rosea, which has pink color flowers. And the third is Vinca oscillate, white flowers with purple center. Next is microscopic characters. Vinca has dorsiventral leaf structure. The upper surface shows the presence of epidermis, which is a single layer of rectangular cells covered with thick cuticle. It consists of unicellular covering trichome and cruciferous stomata. In the mesophyll layer, in the mesophyll region, single layer of elongated and closely packed palisade parenchyma cells are present just below the upper epidermis. Midrib shows the presence of collenchyma below the upper epidermis and above the lower epidermis. The vascular bundles consisting of xylem and phloem is present in the middle of the midrib region, and the rest of intercellular space is covered by 5 to 8 layers of spongy parenchyma. Calcium oxalate crystals are also present. Next is chemical constituents of vinca. A large number of indole alkaloids are present in vinca. Out of them, only 20 dimeric indole dihydroindole alkaloids possess oncolytic activity. Oncolytic activity means anti-tumor activity. And among them, vincristine and vinblastine are most significant. Vinblastine contains indole alkaloid part called catharanthine and dihydroindole alkaloid part called vindoline. The other alkaloids present in vinca are ajmalacine, serpentine, and other serpentine and tetrahydroalstonine. It requires about 500 kg of crude drug to extract about 1 gram of vincristine because it contains very less percentage of alkaloids. This is the structure of vincristine and vinblastine. Both just differ from one structure, that is, vinblastine consists of a CH3 group and vincristine consists of a CHO group. Uses of vincristine. Earlier it was used for soothing and healing of inflammatory ailment of the skin as well as eye irritants and infection. Nowadays it is used to treat diabetes, high blood pressure, asthma and different types of cancer. Vincristine is a cytotoxic compound which is used to treat leukemia in children. Vincristine sulfate has an is an anti-neoplastic agent which acts by arresting mitosis at metaphase. It is given intravenously in the treatment of leukemia. Vinblastine is an anti-tumor alkaloid used to treat Hodgkin's disease. Vinblastine sulfate is also an anti-neoplastic agent and also acts by arresting the mitosis at the metaphase or by interfering the amino acid metabolism. Due to this, the immune response is suppressed. It is mainly used to treat Hodgkin's disease. It is also used to treat hypertensive and diabetes. Dose: vincristine sulfate is given 10 to 30 microgram per kg of the body weight, which is given intravenously, but maximum up to 2 mg. Vinblastine sulfate, it is given 100 micrograms per kg body weight intravenously. The commercial products of Vinca are: first is Vinca powder. It is widely used in the treatment of diabetes, blood pressure, sore throat, and leukemia. The second one is Vinca tincture, which is used in the prevention of various forms of cancer. Anti-diabetic activity, reduces high blood pressure and relieves anxiety. Thank you.
This conference will now be recorded.
What is the biological source of vinca?
Whole right plant of Catharanthus.
Roses.
Yes, yes. Roses, yes.
Main chemical constituents of vinca.
Vincristin and vinblastin.
Over, ma'am. Thank you.